Hey, hello everyone. Welcome to our another webinar series of Glycan Training Association. I am the chair of this webinar, Faiza Chenarbu from SFU. Before I start introducing our speaker, I would like to draw your attention to a EDI survey. Please spend a couple of minutes and fill out our EDI survey. It's going to be a link in a chat soon. Okay, it is my great honor to introduce our next speaker for this webinar, Dr. Professor Kelly McNagny, who obtained a Bachelor of Science degree in Biology and Biotechnology at Worcester Polytechnic Institute in Massachusetts. And he subsequently did his PhD in cellular immunology at University of Alabama at Birmingham. So he then moved to MBL in Germany, where he performed postdoctoral studies in Dr. Graf labs. And now he is currently a full professor at Department of Biomedical Engineering and Medical Genetics at UBC. Dr. McNagny, please take it away.
Thank you very much for the kind introduction, and thanks for inviting me to give the talk. I'm really honored. I'm going to apologize in advance for not talking so much about glycosylation today. I did talk about that at the GlycanConnect conference earlier this year, and I'll tell you instead about some interesting things we've been doing on immune response and development. And it's something that I'm really excited about. And I wanted to share it with you because I think it changes the way you think about disease when you really understand how an immune system is sculpted. So taking it from there, this is kind of the overview I wanted to share with you today. I want to give you a brief introduction on our view of the immune system, tell you about how we view innate versus adaptive immune responses, and then tell you about subspecialties of lymphocytes and how we think different subsets of lymphocytes can polarize an immune response really early in your first encounter with a pathogen or in chronic inflammation. And tell you about, kind of subspecificities with antiviral, antibacterial, and antiparasitic responses. And then I'm going to share with you some new data where we've been looking at how immune response is sculpted from the earliest stages of development by the microbiome, and how that can lead to a very different immune response in you as an adult based on your exposures right after you were born, or even probably before you were born. So I'll tell you a story about Th2 immunity, then I'll tell you about a parallel story where we've been looking at Th17 immunity, and then give you a few crystal ball thoughts on how this changes the way we think about how you're protected from infections throughout your life. So with that introduction, this is kind of the family of portrait of immune cells that we like to think of in my lab. And I'm just going to highlight the fact that for the past 50 years, most people have been laser focused on B cells and T cells, B2 B cells specifically, and alpha beta T cells. And there's a really good reason for that. First, they're the most abundant lymphocytes when you look at adult tissues. And second is that these are the exquisitely specific, antigen-specific cells. If you target them with a vaccine and you get a good response from B2 B cells and alpha beta T cells, you'll be immune to whatever you encountered for the rest of your life. You'll be protected. So those are the cells that we've been studying for about 50 years. But one thing I wanted to highlight here is that if you and I were dependent on these cells to protect us lifelong, we'd all be dead. And the reason for that is that B2 B cells and alpha beta T cells, they take a while to develop. So in order to get really good antigen-specific polarization, it takes you a week to 10 days to get these cells fully revved up. And infections with viruses replicating every 20 minutes or so would quickly overwhelm you. And so what people forget is that we have a whole series of other cells that respond a bit more quickly, but a little less specifically. So on the B cell side, we have something called B1 B cells, and these are the B cells that develop right as a fetal tissue and shortly after birth. They tend to be the cells that make a lot of IgM and not isotype switch readily. And they're broad specificity and low affinity. So they tend to recognize common patterns in pathogens rather than being exquisitely specific. So what good is that? Well, when you're born from that sterile environment in your mom and you come into the world and you get that sewer of antigens colonizing your gut, colonizing your skin, exposure to all these new molecules, you need something that just slows down those infectious agents enough so you can then engage in adaptive response and get these more highly specific B cells. And that's what these B1 B cells do. They recognize patterns with lower affinity. On the T cell side of things, we've got gamma delta T cells, NK T cells, and mate cells, mucosally associated invariant T cells. And again, these cells are lower affinity, broader specificity, and they tend to recognize patterns of antigens rather than specific antigens. They don't even recognize classical MHC. They tend to recognize MHC-like molecules. Okay? If you go further down the scale, you have things like plasmacytoid DCs and innate lymphoid cells and NK cells. These don't even have antigen-specific receptors, so they're quite different, but they fulfill all the same functions: broad specificity, polarizing an immune response for the right type of pathogen, but not quite as specific. Okay, so let's go a little bit further. In immunology, we love to break things down into groups. So the three major groups of responses that I'm going to talk about today are Th1 responses, which tend to be either antiviral or anti-tumor. These are driven by things like interferon gamma and TNF. They're exquisitely attuned to helping you blow up viral factories. They tend to be more T cell and NK cell mediated at killing cells that have been infected, and they make things like IgG and IgA, antiviral, anti-tumor responses. Th2 responses are designed to help you fight parasites, which are these big multicellular organisms, much more difficult to deal with. And so you make a whole series of different cytokines, IL-4, 5, 9, 13. You can think of those as kind of the middle number cytokines. These are designed to make B cells become activated and make antibodies like IgG1 and IgE that bind to these big multicellular organisms, coat them, and drive effector functions from eosinophils and mast cells. It's quite different, okay? On the Th17 side of things, these are mainly for fighting things like bacteria, fungi. They use the high number cytokines. They're designed for activating neutrophils and Th17 T cells and making a different isotype of antibody, which is better at fighting bacteria. So those are kind of the three buckets we throw things into. Now, coming back to those T cells, we have Th1 T cells, Th2 T cells, and Th17 T cells, which make the right cytokines for the right type of pathogens. But we also have these innate lymphoid cells that I talked about just a few minutes ago, which don't have antigen receptors, but they make all the same cytokines as these specific T cells. So three different types of T cells, three different subtypes of innate lymphoid cells. And in fact, if you looked at gamma delta T cells, or you looked at mate cells or NK T cells, you'd find three different subsets of those too. Why is that? Well, that's because these cells sit in tissues. The minute you get an infection, they make the right kind of cytokine for fighting that specific type of pathogen. And we think that these are the cells that guarantee that when you get infected with a virus, you don't immediately start making an anti-parasite response, you make an antiviral response. So they really code the proper response long before you engage in adaptive response. Okay, so now I want to switch gears a little bit and tell you about chronic inflammation. And I want to tell you how I think, or my lab thinks, these immune responses are actually sculpted during development, and how if you don't get the proper education during development, you could be set up for chronic inflammatory disease much later in life. And we're going to start with allergic asthma as an example. So, what do we know about asthma? Well, We know that there are risk factors for asthma, and we know that there are protective factors for asthma. We also know that the rates of allergic asthma have been going through the roof over the past 30 or 40 years. And our genes have not been changing over the 30 or 40 years, but our environmental exposures have been. And there's a hypothesis called the hygiene hypothesis, which suggests that now that we're living in much cleaner environments and we get less exposures to microbiome and environmental insults, we don't have as much infectious disease, but we're getting more chronic inflammatory disease. And maybe that microbiome is educating us to protect us from chronic inflammatory disease. And if you don't get that education because you're in a clean environment, then you're susceptible to this disease. So risk factors for asthma are things like being born by cesarean section or formula feeding rather than breastfeeding, smoking and obesity, antibiotic use. If you use high-dose antibiotics on kids before the age of 2, multiple doses, you're much more likely to be allergic as an adult than if you don't. Interesting, huh? Protective factors, older siblings, if you have more than one dog in the house when you're born, if you have a natural childbirth or breastfeeding, if you live on a farm, if you get a parasite infection as a young kid, these tend to be protective for developing asthma later in life. So all of these things are focused on microbiome. We wanted to study how that actually works. And we started by treating mice with antibiotics to try to mimic those kids who get lots of doses of antibiotics before the age of 2 and seeing if we could mimic that susceptibility in mice. So here's how we did it. We took pregnant mice and we put them on antibiotics right after they got pregnant. And the reason we did that is because we wanted to make sure that when the pups are born, the very first bacteria they encounter are coming from a skewed environment. So we put these mice in sterile cages, we change the microbiome of the pregnant mom, and that ensures that when these pups are born, the first bacteria they see are different from what you would get in a mouse that didn't have antibiotic exposure. We then let the mice develop, and then we run them through a model where we make them allergic to ovalbumin, and we ask, were the mice on antibiotics more or less susceptible to becoming allergic? So here's one of the first experiments we did, and this is about, from 10 or 15 years ago. Here's a normal lung, and you can see the nice airway here with nice epithelia. Here's the lacy alveoli in the lung, and you'll find some blood vessels as well. If we put mice on streptomycin and we make mice allergic, or if we just make mice allergic all by themselves without any antibiotic, we see that you get exactly the same degree of inflammation between those. If we put mice on vancomycin, however, we get much more severe asthma in these mice. So that suggests that some antibiotics, like vancomycin, can make you much more susceptible to allergic disease. And I want to highlight that this is also true of germ-free mice. If you take germ-free mice and you make them allergic, you find that they get much more severe allergic disease than mice that are not germ-free, suggesting that the microbiome actually protects you from developing asthma. Okay, when does that get hardwired? Could you do that in adult, or does it have to be done in young mice? So we did that. We put mice on antibiotics from birth and then took them off antibiotics right about the time of weaning, about 3 or 4 weeks after birth. Or we didn't give mice antibiotics and we started treating them with antibiotics when they were adult and then tried to make them allergic a month or 2 later. If you do that experiment, here's what you see. So no antibiotic and no allergies, you get very few inflammatory cells in the lung. If you make mice allergic to ovalbumin, you get a big increase in inflammatory cells in the lung. If you do that with mice that have been given vancomycin from birth, you get a big increase in inflammatory cells in the lung. Streptomycin, no. Do the same experiment, but now do it in mice that were only given antibiotics as adult. You see no difference. So what that suggests is that changing the microbiome either right at birth or shortly before birth hardwires you to be more susceptible to inflammatory diseases in adult. Pretty amazing result. Okay, what's different about the bacteria dependent on what antibiotic you give? So here we're looking at individual mice, and this color code tells you the different types of bacterial species we found in the gut of those mice. So individual mice have a very broad variety of microbiome, as you can see here from this color coding, if you were looking at control mice. If you put mice on streptomycin, it's a little bit more narrow. You don't have as broad a variety of microbes. Put mice on vancomycin, you get a big shift in the microbiome. And I'm not going to go into great detail, but I just want to highlight that the bacteria you tend to lose with vancomycin are known to be the ones that make things like short-chain fatty acids, things like acetate, butyrate, and propionate. And one of them in particular, butyrate, is a short-chain fatty acid that you really can't make without having the right bacteria in your gut. You as a mammal don't make a lot of butyrate, but if you have the right microbiome, you ferment fiber and you make lots of butyrate. And butyrate is a well-known epigenetic modifier. It modifies gene expression in your genome. People used to use it when I was a kid for trying to induce cell lines to differentiate in a dish, and it works really, really well. So we were particularly focused on butyrate. Okay, are those short-chain fatty acids lower in the mice that we treated with antibiotic? Well, if you look at butyrate, yeah, if you look in the feces or the cecum, you find that those mice have lower levels of butyrate. Acetate and propionate, not so much. Okay. So if everything I've told you is dependent on butyrate, if what we're really depleting with antibiotics is the bacteria that make butyrate, and butyrate is the key, then we ought to be able to give mice antibiotics and add back exogenous butyrate and reverse the susceptibility to asthma. So we tried that. And here's what we see. BAP stands for butyrate, acetate, and propionate. We put that in the drinking water of mice. So here's naive mice, no difference in the inflammatory cells in the lung. If you make mice allergic, of course, you get more inflammatory cells. If you add butyrate and propionate and acetate, doesn't change. That's in regular mice. On the other hand, if you put mice on vancomycin, again, you get much more severe asthma, more inflammatory cells. And if you add butyrate to the vancomycin, now you bring those mice right back down a register with the wild type mice that never saw antibiotic. So what I'm showing you here is that probably the key to protecting you from getting allergic is a biome that produces butyrate. And if you add butyrate back in trans without the bacteria, you can suppress development of asthma as an adult. Pretty striking. Okay, so quick summary of some of this old data. We know that antibiotic shifts in the gut flora increase your sensitivity to developing Th2 disease. We know that this coincides with the microbiome that produces short-chain fatty acids. If you lose that microbiome, you're more susceptible. If you get back the short-chain fatty acids in trans, you can reverse the effects of that antibiotic. And altering the microbiome early in life can have a long-lasting effect on your susceptibility to disease as an adult, which I think is really striking. And I want to point out that that data I've shown you now in mice is backed up by what we know in human. If you look at kids in a big birth cohort, which we have in Canada, and you look at their microbiome at birth and 3 months after birth and 1 or 2 years after birth, you find that kids who don't have a very diverse microbiome within the first 3 to 6 months of birth are much more likely to develop asthma by the age of 5. So it looks like this correlates very well with what we see in our mouse model. Okay, so that's what we knew as of about 10 years ago. The question then becomes, okay, what is really that microbiome and butyrate doing at the mechanistic level? How does this all work? So here's how we think of how an allergic response really works. Almost every allergen is a protease. If you look at house dust mite, the thing that 85% of people are allergic to, there's a protease in house dust mites that damages epithelia. That damaged epithelia immediately releases these alarmin-type molecules, things like TSLP, IL-33, and IL-25. These are alarmins that are instantly released from the epithelia. They activate innate lymphoid cells, which live just below the epithelia. Instantly, those innate lymphoid cells start making IL-5 and 13. IL-5 recruits the eosinophils within hours of first exposure, long before you've had time to make an adaptive response. IL-13 activates dendritic cells. Dendritic cells then grab any antigen they find in this local milieu. They run to the local lymph node, and now they begin to prime T cells. And they polarize those T cells to making T cell receptors against a very specific antigen and to make Th2 cytokines. Those things then activate B cells to isotype switch and make antigen-specific B cells. This is about a week after you've been exposed to an allergen. Now these cells all come out of the lymph node through the draining lymph. They migrate through the blood back to your lung. And now you're going to be allergic for life because you've developed antigen specificity to specific antigens that cross this epithelial barrier. Everything before this was antigen non-specific. Okay. So the question, are any of these cells altered by our treatment with antibiotics? Do we hardwire anything different about these cells? Now, just to show you first that these ILCs are critical for developing allergic responses, here we've taken some genetically engineered mice where we've knocked out ILCs, ILC2s specifically. And if you look at the lungs of those mice where we've made them allergic, here's wild type mice. You see lots of inflammation in the lung. When we expose them to house dust mite, that antigen that 85% of people are allergic to. Here's mice where we've deleted the ILC2s, and you can see they have much less inflammation. So the ILC2s are critical for developing allergies. That's true of the ILC2s. Here's what happens if you look at eosinophil recruitment in the lung. With ILC2s, you have lots of eosinophils in the lung. Without ILC2s, you have very few. If you look at cytokine production when you delete the ILC2s, first, the number of cells that you develop are down. So you have fewer total cells in the lung when you delete ILC2s. The main difference is you have fewer eosinophils. If you look at the adaptive responses in those mice, they have much less IgE specific for house dust mite if you get rid of the ILC2s. So even the downstream adaptive response is dampened if you get rid of the ILC2s. If you look at cytokine production, IL-4 is down, IL-5 is down, IL-13 is down. All those Th2 cytokines are lower in mice where we delete ILC2s. So this all says that ILC2s are critical for getting a good allergic response. So quickly paraphrasing, this is the pathway for developing a Th2 response. If you delete ILC2s, the immediate response is not there. You don't recruit eosinophils and you don't activate DCs. Downstream from that, you don't develop Th2 T cells. You don't develop IgE. Everything is dampened if you get rid of ILC2. So they're pivotal in developing an allergic response. Now, coming back to that question I posed a few minutes ago, are any of these components altered if you give antibiotics early in life? Do we amplify any of these cell types? So here's the first foray into looking at that. We started by looking at ILCs. Now here I'm showing you FACS profiles where we're looking at the frequency of ILC2s in the lung of mice that we were either given antibiotics or not given antibiotics. The way you recognize ILCs is you stain for all of the lineage markers for T cells, B cells, monocytes, and macrophages. You look for the cells that are negative for all those markers and positive for IL-7 receptor. Here are the ILCs, 1s, 2s, and 3s. Here we're gating for the ILC2s specifically. And in the mice that got antibiotics, vancomycin, we see twice as many in the lung at steady state. These are mice we haven't done anything to except given them antibiotics in the drinking water. At steady state, they have twice as many ILC2s as mice that didn't have antibiotic. If we look at those ILC2s and what they make, here we're staining for intracellular IL-5 or intracellular IL-13. Mice on normal drinking water make a little bit of those Th2 cytokines. Mice that we gave antibiotics without any perturbation at all are making high levels of those cytokines. So at steady state, they're making more of the inflammatory cytokines even before we make them allergic. They also acquire the ability to make a new cytokine, IL-2. So this is showing that they've actually gained an ability to make things that we normally don't see very much of in ILC2s. Okay. So far what I've shown you is that if you give mice antibiotics, you have more ILC2s, twice as many, and at steady state, they're making more cytokine. What happens if we start inflaming the lungs of those mice? Okay, so we took our mice, we gave them antibiotics or not. We put intranasal IL-33, that alarmin that activates ILCs, to see what happened in the lungs right after that. And this is within a few hours of treatment. When we look at that, what we find is that we get a huge increase. So here's that steady state. Here's after induced dysbiosis with priming with IL-33. We see a big jump in the amount of ILC2s. They expand really robustly. And they also acquire the ability to make a new cytokine, IL-4. Without antibiotic treatment, they don't. With antibiotic treatment, they make huge amounts of IL-4. IL-4 is a Th2 cytokine that lung cells normally don't make unless they've been exposed repeatedly to an inflammatory cytokine. On the very first exposure, we see these cells are pumping out lots of IL-4. So I've shown you that we have more ILC2s at steady state. Now what I'm showing you is that when you activate those ILCs, they make a new cytokine, IL-4. What's downstream from that? Well, downstream from that, we know that IL-4 activates B cells. And IL-4 induces B cells to make an isotype switch to making IgE. That's the antibody that comes up when you get allergic. Very specific for allergic disease or parasite infections. So we looked at IgE production in our mice. And what we found was that if the mice had been exposed to antibiotics, they had really high levels of B1 B cells. These are B1 cells, here are B2 cells. Really high levels of B1 B cells that were making IgE. And this is non-specific. It's just IgE, not antigen specific at all. And this is just shown graphically here. The cells are making huge amounts of IgE before we've even made them allergic. So there's an increased production of natural IgE. And I'll remind you what I told you in the very beginning. B1 cells are pretty antigen non-specific. They made broad specificity antibodies. So presumably these are making antibodies that will bind a lot of different antigens. Okay. So we've got more ILC2s. They're making more cytokines. They're making a new cytokine, IL-4. That induces isotype switching of B1 B cells. These cells are now making IgE before they've even been exposed to a parasite or an allergen. And that is in the circulation of the mice. What's downstream of IgE production? Well, downstream of IgE production are basophils and mast cells. Basophils and mast cells have a Fc receptor for IgE. They bind it, it coats those cells, and if you've ever had an allergic attack, you know that you've got these mast cells sitting in your tissues, and you cross-link IgE on them, and they start to release histamines, and that's what makes you kind of start crying and getting mucus and sneezing and wheezing. That's all due to histamine release from basophils and mast cells. If you look at the IgE stuck to basophils and mast cells in mice on regular drinking water, they have a bit of it bound. If you look in the mice that were treated with vancomycin, they've got 10-fold more IgE coating their basophils and mast cells than the normal drinking water mice. So you've got cells that steady state coated with IgE, and it's that IgE that comes from the non-antigen specific B cells, the B1 cells. So presumably anything that crosses that epithelia when you get damage will cross-link the IgE on those cells, and they'll start releasing histamines and start making you feel allergic. So we've got an expansion of eosinophils. We've got an expansion of B1B cells that are making IgE, which they normally should not make. That IgE is coating mast cells and basophils. This now makes you prone to an allergic attack. Okay. If we look at those antibiotics, I'm going to go through this quickly. The question is, I've shown you at the very beginning that we think that the antibiotics are removing the bacteria that make short-chain fatty acids. I showed you we can reverse allergic susceptibility if we give those short-chain fatty acids in trans. What if we give antibiotics plus short-chain fatty acids to our mice and we look at the ILC2s and we look at their IL-4 production? If we do that, I've shown you that without antibiotics, you get an increase in ILC2s and IL-5 production. If you put mice on vancomycin, that's amplified. If you give back short-chain fatty acids and trans, it brings them right back in register with the mice that were on normal drinking water. So we reverse that expansion of ILC2s. We reverse the production of IL-13, IL-5, and IL-4. So we can reverse everything I've shown you about the allergy susceptibility in that pathway just by giving short-chain fatty acids in the drinking water. Okay, this is just summarizing what I've told you. This is the pathway that gets amplified if you've been on antibiotics as a kid, and we can reverse all of that with short-chain fatty acids. And if you want to read more details about that, we did publish this at the very end of last year in December 2024. It's a great paper. I encourage you to read it. Okay. Everything I've told you about is applicable to Th2 disease and Th2 inflammation. What about Th17 disease? Now, we're not talking about the bacterial responses or the asthma responses. Let's talk about something different. Let's talk about Th17 disease. This is prone for bacterial infections, but the other thing that Th17 responses do, those are the responses that are pathological when you get things like Crohn's disease and gut inflammation and gut fibrosis. They're the responsibles that drive multiple sclerosis. They're the responses that drive development of rheumatoid arthritis. Are those influenced by early exposure to microbiome too? Now, when we started this project, we thought, okay, if you're more susceptible to Th2 disease, then maybe you're protected from Th17 disease. So maybe it's a seesaw, one drives more inflammation and you're protected against this other type of inflammation. The other possibility we thought about was, well, maybe antibiotics like vancomycin, maybe they make you susceptible to any inflammatory disease. It's broad spectrum. So if you get rid of the microbiome that makes short-chain fatty acids, maybe that'll make you more susceptible to Th17 disease too. Turns out we didn't see either of those things. And I'll show you what we saw when we did this kind of a study. So we've got a really good model for Th17 lung inflammation. It's a disease called hypersensitivity pneumonitis. It's a disease that people call occupational asthma. Things like farmer's lung, bird fancier's lung, hot tub lung, sausage worker's lung, great names. These are all lung inflammatory diseases that are driven by Th17 inflammation. And a classic example is in Quebec, you've got farmers who get allergic to something. They get something called farmer's lung. They walk into a barn with moldy hay. They get exposed to a bacteria, causes them to get this really bad lung inflammation. They go away, they feel better. They come back to the barn, they get it again. It's not Th2 driven, it's Th17 driven. It's a neutrophilic inflammation, very different from asthma, and it's mainly T cell driven. There is no therapy for this disease. The only therapy is to walk away from your job and get a new job and get away from the barn. We can mimic that disease exactly in mice using the same antigen that drives the disease in human. So here's the experiment. We've put mice on antibiotics again, and we've asked, are they more susceptible to this Th17 lung inflammation? Here are the lungs again with no treatment. Here's exposure to that antigen that gives them farmer's lung. They get a lot of inflammation. Look at the vancomycin, no difference now. It's exactly the same as the wild type mice. But here's the mice that got exposed to streptomycin, the different antibiotic. They're more susceptible to this Th17 disease. So different antibiotics activate and hardwire different pathways to make you susceptible to disease. This is showing that data graphically. So here we put the mice through the farmer's lung model, no inflammation. Here we're making them in the model. They get much more inflammation. With vancomycin, it doesn't change. With streptomycin, it's much worse. Very different from the Th2 disease. Here's the cytokine data. Give the mice farmer's lung, you get more inflammation, you make more interferon gamma. Doesn't change with vancomycin, but with streptomycin, it's much worse. IL-17, the classic cytokine for Th17 responses, no difference with vanc, now much higher with streptomycin. Okay, so does streptomycin alter the pattern or the frequency of ILC2s in the lung in a way that's different from vancomycin? And the answer is yes. So we put the mice through the farmer's lung model. We again look at the ILCs. These are all the ILCs, 1s, 2s, and 3s. We're looking at expression of a transcription factor, Rho gamma T, that's very specific for ILC3s, or TBET, a transcription factor that's very high in ILC1s, so 3s versus 1s. And what you see is that we get a big increase in the ILCs that express Rho gamma T, i.e. the ILC3s. So now we're not seeing more ILC2s, we're seeing more ILC3s in the lung. And again, it's a doubling at steady state in the mice that were exposed to streptomycin, but not vancomycin. Okay, what do those lungs look like after priming? Again, we're doing the same experiment I showed you with asthma, but this time with the Th17 antigen. Here's the mice where we've done that. Here's the normal drinking water mice. Here's the mice that were on streptomycin and activated with SR. And you can see that you get a huge increase in the Rho gamma T positive ILC3s over the normals, and a big increase in ILC1s now once you start driving the inflammation. If you now look at the production of IL-17 from Th17 cells or ILC3s, you see a big increase in IL-17 production. So once again, treatment now with a different antibiotic expands the frequency of ILCs, this time it's ILC3s, and it primes it for making more IL-17. We also see a big difference in the antigen-presenting cells. So if you activate those mice and you take a look at the degree of macrophages and dendritic cells in the lung compared to normal biotic mice, we see a huge increase in the number of antigen-presenting DCs in the mice that got streptomycin. That's shown graphically here. Let's get a little bit more granular. So we know that Rho gamma T is a transcription factor that's absolutely essential for the development of Th17 cells and ILC3s. Without it, you have mice that have no ILC3s or Th17 cells. Now, one thing that we got really excited about, we were trying to figure out what is driving the expansion of these cells in the mice that we gave streptomycin to. And again, we didn't have short-chain fatty acids driving this. We knew that didn't make a difference. Was there a different bacterial metabolite that we need to drive the expansion of these cells in the mice that got streptomycin? And this is where reading the literature really pays off. So my student was having a look at a paper that had recently come out in Nature, and it was a fascinating paper on bile acids. So some of you may know that every time you eat a meal, you make tons of bile acids, and they get secreted into your gastric milieu. The bile acids help you solubilize food so you can digest it easily. But once they get into your gut, your microbiome modifies the primary bile acids into an entire pharmacy of secondary bile acids. So you modify bile acid into a whole bunch of different things. One of those is a bile acid called lithocholic acid or isolithocholic acid. That gets made in huge amounts if you've got the right microbiome for modifying it. And what these researchers had shown was that patients with Crohn's disease lack the bacteria to modify this bile acid into iso-LCA. So normal people have lots of iso-LCA in their gut. People who have Crohn's disease don't have iso-LCA, and it's due to a lack of a microbiome. So my students saw that data and said, let's take a look and see if that could be the driving force that activates ILC3s. Here's the pathway for making iso-LCA. And again, these enzymes come from the microbiome. If you don't have them in your gut, you don't make iso-LCA. And what these researchers in the Nature paper had shown was that that transcription factor, ROR-gamma T, is inhibited by iso-LCA. So first, we confirmed that we could see that. So here's a ROR-gamma T transcription assay. We showed that if you add iso-LCA, it inhibits ROR-gamma T. It doesn't affect a closely related transcription factor called ROR-alpha. If you now look at what that does to Th17 cells and ILC3s, we could show that you still have ILC3s and Th17 cells, but they're less frequent and they don't make IL-17. So when you make this bile acid, it turns off the production of IL-17. If you don't make that bile acid, it's amplified. Can we add that bile acid back in trans and now repress the ILC3s? So here's where we did that. We made them isoallergic again, or drove them to get hypersensitivity pneumonitis. We see the ILC3s are amplified. If we now add the iso-LCA, we see the ILC3s drop right back down to normal levels. We also show that the frequency of Th17 cells or ILC-17 producing ILC3s drops right back to normal register. So we have fewer ILC3s and they don't make IL-17 anymore. Okay. All the enzymes you need to convert lithocholic acid to iso-LCA comes from the microbiome. So we gave mice this precursor, LCA. We asked, can that inhibit? And we showed that, in fact, that precursor has no effect on suppressing ILC3s. And we showed that actually, if you look at the production of iso-LCA in the gut and the fecal matter from those mice, we've got iso-LCA, but they are unable to convert that to iso-LCA if they were on streptomycin. They're perfectly capable of producing it if they were mice weren't given streptomycin. So you can make iso-LCA if you've got the right microbiome. You can't make it if you treated mice with streptomycin. It's an inhibitor of ROR-gamma T and ILC3s. So that's why these mice are no longer able to inhibit the production of IL-17 and they have an amplified response. So summarizing this point, streptomycin-induced dysbiosis sculpts a completely different hyperactive peripheral Th17 response. This leads to increased priming of tissue-resident ILC3s in the lung. They're primed for inflammation. This appears to be due to loss of the inhibitor of ROR-gamma T, this bile acid that you require the microbiome to produce. Without it, the brakes are off and the ILC3s are hyperactive. In contrast to vancomycin, which sculpts a Th2 response, streptomycin can induce a Th17 response. But what's interesting about this model is you can add the iso-LCA to the adult mice and repress the disease. Why that's interesting is that I've told you that multiple sclerosis, Crohn's disease, hypersensitivity pneumonitis, rheumatoid arthritis, they're all driven by Th17 inflammation. Now that we know the right bile acid we can give back, we might be able to suppress all of these diseases with this microbiome-derived metabolite. And it gives you a pathway for trying to treat a lot of these really hard to treat inflammatory diseases. So that's a summary for the Th17 part. This is just an overarching summary for all of you folks. There are a range of unexplored lymphocytes other than just the traditional T and B cells that we think protect us from reactive chronic inflammatory disease. And I'm highlighting ILCs here. These cells are really long-lived. They colonize your tissues early in life, and they sculpt in a long-term immune response. And that long-term immune response can make you more or less susceptible to these chronic inflammatory diseases. Distinct types of microbiome have remarkably selective effects. Altering one type of microbiome can make you more susceptible to Th2 disease. Altering it in a different way makes you more susceptible to Th17 disease. And balancing the development of these cells early in life is key to protecting you from future dysfunction and disease and chronic inflammation. And if you think about it this way, that kind of tells you that, a lot of these diseases that we think about as old age diseases really started from getting the wrong exposure early in life, and it rethinks the way you want to try to target a disease. If you're a kid who's been exposed chronically to lots of high-dose antibiotics, maybe you want to give a probiotic or a metabolite to lessen your chances of getting disease as an adult. I am not going to be the person to say don't give kids antibiotics. Antibiotics are life-saving drugs. But if you do have to take antibiotics as a kid, maybe you should give a secondary metabolite in addition to the antibiotics as a way of lowering your possibility of getting a chronic inflammatory disease later. Everything I've told you today, really, especially the latter part of the talk, was done by this one really great graduate student, Ahmed Kabil. He's been a godsend. He's a super bright kid, does a lot of reading and ran all the models together with Mike Hughes, as well as a lot of select people, Maggie Chopra and a few other people from my lab who are all participants in this work. I've got a really great lab. You can see here that we are a very diverse lab. I think that's very important. Some great collaborations with Brett Finley's team many years ago, who basically we did the early antibiotic work with to show what increases the susceptibility when you give high-dose antibiotics. A lot of collaborative work with Fumio Takei to look at ILC function. And again, support staff from our transgenic facility, gene typing facility, antibody coupling facility. Really, all our core facilities have been instrumental in all the work I showed you. And I'll just leave you with this lab motto of ours: is powered by science and strengthened by diversity. I've been incredibly lucky in having a diverse lab with people from all over the world. They bring really interesting perspectives on our work, and I attribute that to being the success in my lab. And I think it's really important to highlight that, especially with what's going on south of the border in the US. It's great to have people from around the world. They really enhance our research, and I want to acknowledge that. And with that, I will stop and be happy to take any questions you might have.
Thank you so much for the fantastic talk. So we would have time for a couple of questions. Can unmute yourself and speak up or just chat. Just write
So
it.
I will jump in, perhaps. Sam Abraham. Kelly, some years ago, I was made aware of something that I must admit being entirely ignorant to. And it was the fact that mother's milk actually produces long chain sugars, which we as humans can't do anything about, but it's produced in mother's milk to actually encourage certain bacteria to grow. So this is a co-evolution, if you like,
Yep.
of us and these creatures that make us up. I was fascinated with that, and then went on to discover that there were knock-on effects if you came from a family that didn't give you mother's milk. You could then have problems later when you had your own children.
Yeah.
So there were generational issues associated with it. But thanks for a fascinating talk. Are butyrate and things used currently to mitigate responses?
Currently not, and I think it's something that people should look at. And you're absolutely right. Formula-fed kids are much more likely to develop allergies later in life. They're about twofold increase.
Yeah.
And then you can throw in the pets, that's another twofold increase. So, and cesarean birth is another twofold increase. So these things tend to stack up and make you much more likely to develop allergies. The milk oligosaccharides, absolutely right. They are designed to help you develop a microbiome that's good at making short chain fatty acids and protecting you.
Yeah.
So all of these things go into the mix. And again, we've tended to think of microbiome as being something to be wary of. It's really something you essentially need. The metabolite folks know this, right? If you don't have a healthy microbiome, you don't digest food well, you need a biome in order to
Yeah.
help these things.
Thank you.
Yeah. Others.
Other questions?
I think the thing for us that was really remarkable is the fact that this hardwires your immune response to be pro-Th17 or pro-Th2. And for the asthma stuff, you kind of have to add the short chain fatty acids like butyrate early because it's an epigenetic modifier and it hardwires the cells very early, and you can't change that if you do it as an adult. You add short chain fatty acids as an adult, doesn't have an effect. But with the Th17 disease, I've shown you that bacterial metabolite will turn off Wargema T in adults. So that gives you the opportunity to intervene with those people who have Crohn's disease, who have psoriatic arthritis, or potentially multiple sclerosis and things like that. And I think that's a fascinating opportunity. Sorry, Kirk, you had a question too.
Yeah, no, Kelly, I mean, amazing. It's given me a ton of food for thought. Working mostly in the gut and with the factors that influence host microbial interactions, particularly at the interface. One question I had, I'm not sure, maybe if I missed it, is like, what do you think is the most proximal event happening when you have the metabolites being produced by the bacteria, for example, butyrate?
Yeah.
Where in the gut is it interacting with the immune cell? Is this going systemic and everything? And what receptor? And are the immune cells actually, like, is it direct interaction with butyrate or is it indirect through another mediator?
It's a great question, Kirk. I think that the butyrate probably goes systemic. And the reason I say that is, and this is interesting too, when do allergy susceptibilities really start? So I've shown you that the microbiome can influence it. And everybody's been laser focused on, those few first weeks after birth and the 6 months after birth. But think about that for a minute. Whenever you look at a microbiome and you look at a kid, I would argue you're actually looking at mom's microbiome during pregnancy.
Mm-hmm.
It passes through the birth canal. They get coated with bacteria. That's the first bacteria that goes in their mouth and they swallow. When they breastfeed, they're getting the mom's microbiome. So all of it's coming from your mom.
Mm-hmm.
Is there data to suggest that actually it's during pregnancy when you hardwire the immune response? And I think that's possible because butyrate does cross the placenta readily. And that's why I say, it's a systemic effect. It can go into all the tissues.
Mm-hmm.
ILCs are specified and made during fetal development and shortly after birth. They colonize your tissues during that window, and then you don't make them, they don't circulate after that. They stay tissue resident and they expand and contract in the tissues in response to inflammation. So they're long-lived immune cells, and I think that specification really early determines what happens late. With regard to the gut, ILC3s are the major colonizers of the gut. There are R2s there, but they're more in the intestine, a little bit less in the colon. Lung, it's almost pure ILC2s, unless you have something drastic happen. Then you start getting more 3s and 1s. So they kind of have different frequencies in different tissues. I think that gets hardwired very early. The metabolites do circulate, so they could be exposing them continually and bathing them in different, signals, if that helps.
Mm-hmm. Very much. Thank you. That makes a lot of sense, actually.
Okay, great. Other questions?
I just have a comment. I love you guys. I have a project aboard the ISS, and I'm in DC and HHS. So hey, we're not all that way. Thanks.
I
Thanks very
know
much.
you're not. I know.
Oh, yeah, well, it's okay. It's a good. I just think that maybe we should become the 11th province. I've been to every province and two of the three territories. I love Canada. Oh,
I,
Canada.
I'm
Okay.
American. I'm American by birth. I would recruit you in a heartbeat. It's great.
Yeah. Yeah, you guys got really nice national parks too. Thanks.
Sure.
Okay. Other questions, comments?
Nobody has, to my knowledge, has looked at those lectin receptors and Cglex and other things that would help these things migrate to different tissues during development. I think that would be fascinating to look at, you know. What kind of carbohydrate receptors help these cells migrate? What kind of receptors activate them? That's something that's relatively unexplored, largely because these cells are hard to find. You know, to me, it's always amazing that you could have a subpopulation of lymphocytes that nobody discovered until 10 years ago. I mean, ILCs were discovered 10 years ago. How could you miss them? And the answer is that they're tissue resident, you don't see them in the circulation, and they're rare but super potent. So I think that's just a good lesson in how much we still have to discover in immunity. Love to see somebody trying to figure out how these cells migrate to tissues and how they take up residence in different parts of tissues. For the glycobiologists.
Yes, thank you so much. Please join me and thank again Kelly for the fantastic talk. And thank you all of you for joining us today. Please make sure that you fill out our EDI survey. The link is in the chat. And we hope to see you again in our next webinar, which will happen in September. Thank you.
Thank you.
Thank you. Awesome talk.
Thanks, Kirk.